Hello everyone, I am excited to introduce stem cell application and how stem cells can actually be a concern in cancer development and specific tumor progression. To start off, I just wanted to introduce what a stem cell is just in case any of you guys needed a refresher. A stem cell is an undifferentiated somatic cell that has the capability of giving rise to specialized cells upon division or going through a self-renewal process. These images is something we have previously seen this semester, as you can look at the different pathways a stem cell can undergo upon dividing. Upon division, the stem cell differentiates into another stem cell and a cell that will then specialize in a specific tissue like the ones listed here on the slide. There are different levels of potency that stem cells are categorized in, such as pluripotent, multipotent, and totipotent, which all have different capabilities in the body to replenish damaged or dying cells. Just to give some history, the first placental blood transfer, which is where embryonic stem cells reside, was reported in 1939. Years later, the same application was used to treat a young child with leukemia after chemotherapy treatment to enhance a suppressed immune system. These stem cells are hematopoietic stem cells, which have the potential to differentiate into all types of blood or immune cells like white or red blood cells. These cells are also included in the renewal process of blood. Embryonic stem cells in medical application is showing promising results as the cells are more tolerant to the human leukocyte antigen, which are known as HLAs. In typical transplant, matching HLAs are important in order to avoid graft versus host disease. However, new findings are starting to indicate that unmatched HLAs in the umbilical cord blood transplants, abbreviated to UCBT, from unrelated donors are still showing promising engraftment results. UBCT are used for treatment of malignant diseases such as acute leukemia and lymphomas, and non-malignant diseases such as inherited metabolic disorders, which are typically a result in a single gene dysfunctional enzyme. However, these stem cells can also be used in other diseases such as heart disease or type 1 diabetes. One fun fact I wanted to share with you guys is that if an individual suffers from a heart attack and survives, these stem cells could be essential for replenishing damaged dead heart cells. As our professor has previously talked about cancer stem cells, I wanted to bring up the underlying concerns during cancer therapy. Cancer stem cells can originate from a variety of places, such as stem cells that have proto-oncogene activation, progenitor mutation, or even a normal cell that has become cancerous and taken on properties of stem cells. Cancer stem cells are a subpopulation of cancer cells that could be the reason why patients relapse. These cancer stem cells, after being exposed to chemotherapy treatments, can cause heterogeneity in tumors. This could be the key reason why certain drug therapies fail and patients relapse over time. I just wanted to give you guys a figure to really put into play the pathways that cancer stem cells have the ability to undergo. These pathways are all contributing factors to why CSCs are resistant to some chemotherapies. Topics that we previously talked about throughout this course are concerns when dealing with cancer stem cells. In cancer, the epithelial mesenchymal transition and stem cell markers are co-expressed in the circulation of tumor cells with metastasis. EMT activation can increase drug resistance and induce invasion and tumorigenesis. Other pathways such as resistance to DNA damage, induced apoptosis, signaling pathways, epigenetic mechanisms, tumor environment such as hypoxia, and inflammation are all contributing factors to the complexity of curing cancer. The first study I wanted to introduce to you all is one that deals with the use of umbilical cord blood and NK cells' effective chimeric antigen receptor, CAR, therapy against CD19 positive leukemic cells. I wanted to briefly go over the testing subject of this case study that used primary chronic lymphocytic leukemia cells from 6 patients for in vitro studies of NK CAR functionality. The goal of the study was to evaluate the efficiency of CAR NK cells from different cell sources against CD19 expressing leukemic cells. For the purpose of this study, I do want to point out the use of interleukin-2, which was used to increase NK cell population, as well as interleukin-15 to help with survival, proliferation, and higher cytotoxicity from both sources. The interleukin family is expressed in many inflammatory autoimmune diseases and can be essential in our immune response to therapies like this one. Some results of the study focuses on CAR-altering specificity and function of T cells in our immune response. These results can be hard to understand unless you have time to read the entire paper. However, we can see how CAR T-cell therapy can be successful in targeting hematological cancer targeting B lymphocyte CD19 malignancies. Moving on to the second clinical trial, the article studied glioblastoma multiforme, which is the most aggressive brain tumor in adults. The typical treatment for this type of tumor is surgical resection, but often cannot be implemented due to the location and risk factors that align with this operation. This article's purpose was to really understand the importance of immunotherapy's ability to target glioblastoma stem cells, which is one of the reasons why glioblastoma multiforme, GBM, is resistant to conventional therapies. These researchers used HER2-specific glioblastoma multiforme cells by transferring genetic material inside a cell via retroviral vectors for HER2-specific chimeric antigen receptors. The effector activates HER2-specific T cells against autologous glioblastoma multiforme, which was done in vitro. One result from the study was that stimulation of HER2-specific T cells when HER2-positive analogous GBM cells were present, T cell proliferation increased. HER2-specific T cell killed CD133 positive and negative cells from the original HER2-positive GBM, but the HER2-negative tumor was not killed. T cells in patients with GBM can be modified with the human epidermal growth factor receptor 2, which is responsible for producing effector cells, which is important for immunostimulatory cytokines to kill primary HER2-positive tumor cells, which are CD133 positive and GBM stem cell positive. These results conclude that gene transfer for the generation of HER2-specific T cell generation from GBM patients could be promising for anti-tumoral activity against tumors with HER2-positive receptors. This could also be effective for GBM stem cells, resulting in the immunotherapy treatment of HER2 redirecting T cells for GBM patients. The last study I wanted to introduce focuses on stem cell antigen CD133 as it is identified in tumorigenic populations in glioblastoma patients. CSC cells are hypothesized as being one of the main components of tumorigenic cells in tumor development. In order to conduct this study, they used 44 glioblastoma patients that were treated with complete or partial tumorectomy, followed by radiotherapy combined with this drug listed here on the slide, in order to measure the death or disease progression. These researchers were looking for a correlation of tumor formation with CD133 positive. This drug prevents the cell from making DNA, preventing mitosis from occurring, and decreasing proliferation like seen in the figure above. However, this drug does have side effects which are not limited to nausea, vomiting, loss of appetite, and insomnia, all which could suppress the immune system. The main finding of this study really represented that the patients with glioblastoma with a greater than 2% expression of CD133 positive cells directly correlated to an increased amount of cancer stem cell and progenitor cells. This correlation did not suppress disease progression and allowed for tumor formation. One conclusion we can make is that when cells have HER2-positive receptors, modification and inhibitory therapies could most likely have a higher impact on preventing tumor development in GBM patients. As discussed in the two studies, when CD133 and T cell recruitment was increased, the demand of proliferation also increased. As these two studies were able to find correlating results, the next steps researchers could take is implementing a treatment of inhibitory proteins or antibodies that have minimal side effects for HER2 receptor-dependent tumors. CAR therapy can also show promising results for many types of cancer as it activates T cells by receptor redesign, which triggers proliferation. The new T cells can then recognize specific antigens like these studies focused on and can be used to attack those invading cells. Secondly, cancer stem cells have the ability to proliferate and divide and specialize in a variety of tissues and cells. We see now that higher abundance of CSCs cause heterogeneity and increase the durability of tumor cells against treatment. These studies show how impactful these cells can be, especially when present with HER2 and CD133 positive. These are now 3 things researchers would have to take into account when trying to treat and develop treatments for patients with similar tumorigenic cells. Lastly, as chemotherapy is a high therapy on the market, there are high side effects with many combination drugs. Clearly, one of the first goals would be finding out what demand these cancer cells require for cell proliferation in the first battle for the in vitro step. Researcher needs to find less intensive therapies. Triggering an immune response and inflammatory cytokines could also progress the development of cancer and could be the cascading effect on this disease. Chemotherapy is very expensive, and as these drugs are the impacting reason why therapies are working, doesn't mean that everyone has access, which is the biggest issue that I see, since everyone should have access to these kinds of treatments for remission. We still have a lot to learn about the treatment variability of the many different types of cancer, but hopefully researchers soon can continue to make an impact on decreasing the progression of these infectious cells.